Funding for this program is provided by Annenberg CPB to advance excellent teaching.
In 1959, researchers believed they'd finally delivered the answer, proof that the Y chromosome determines male sexual development in humans. But is it just the Y, or is there something more complicated at work? And if the Y makes a male, then what makes a female?
Biologists have been saying for half a century, female development is a default outcome. I don't think the available data supports this idea.
And even if science can determine we're male or female, how do we know, deep inside, what sex we are? Researchers are providing some surprising answers and some even more perplexing questions as we explore the biology of sex and gender. Okay. 7 weeks after sperm meets egg, something interesting happens. Chemical processes take place that determine whether the undifferentiated gonad in the embryo becomes testes or ovaries, male or female. And from that point forward, a delicate dance takes place inside our bodies as genes and proteins play their part to create our male or female body. How does it happen? What are the processes that make that decision, and how is that decision affected to carry out the development of male or female? And what about gender, our own sense of being male or female? Where does it come from? The ability to explore sex and gender at the molecular level has revealed some of the key genes involved in sex determination and answered some questions. But as in all areas of biology, some of our questions may not have simple answers. In the early 1960s, Soviet track star Irina Press and her sister Tamara came under suspicion from the International Olympic Committee. Though the committee had no proof, they suspected the sisters were actually men passing as women in the competition. So the committee began testing all female competitors. Originally, athletes were required to walk naked in front of a panel of judges, but the athletes complained that the procedure was humiliating.
In 1968, the International Olympic Committee decided to not do any physical examinations anymore, but to leap into the modern science, and they started looking at chromosomes. And it was decided that XX athletes were female and XY athletes were male.
Women whose karyotype showed a Y chromosome were ejected from the competition. One of the athletes who failed the test is Maria Patino, the Spanish national junior champion in the women's hurdles. Patino has androgen insensitivity syndrome. Even though she has a Y chromosome and her body produces more testosterone than most women, she lacks the receptor protein that cells use to detect testosterone.
The inability for testosterone to bind to its receptor make the external appearance of these individuals to be fully female.
Eric Villain is a researcher and physician at the UCLA Medical Center in Los Angeles.
They do not have a penis. They do have what appears to be a normal vagina. They just look normal female at birth.
Each year, 1 in every 13,000 babies born with a Y chromosome has androgen insensitivity syndrome. The condition doesn't give these females any added strength or endurance.
Even the fact that their levels of testosterone is high, it does not help them in terms of muscle mass as the Olympic Committee could be afraid of, because these high levels of testosterone have no effect.
In fact, there are many conditions in which a person's physical appearance does not match their chromosomal makeup. No one knows how many of these individuals were prevented from participating in the Olympics. But Maria Patino fought back, challenging the decision. Even with her Y chromosome, she argued, she is still fully female. 3 years later, she was reinstated. And in 2000, the Olympic Committee abandoned routine sex testing. So where did the Olympic Committee go wrong? How did answering such a fundamental question get so complicated?
That Olympic Committee had a desperate need to have a simple definition of what it is to be male or to be female. And I think we know from our own personal experience that there is no such thing as a simple definition. And even within the scientific context, sex or gender has been defined at many different levels.
Oh, hi, Dr. Page. How are you?
What you got?
David Page is a researcher at MIT's Whitehead Institute in Cambridge, Massachusetts.
Who makes a different kind? The classic formulation now in humans or in mammals starts with a sex chromosome definition. Are you XX or are you XY? The classic formulation then moves on to a definition which looks at the gonads. Do you have testes or ovaries? And then moves on to the internal accessory structures. Do you have a uterus and fallopian tubes, or do you have an epididymis and vas deferens? And then moves on to the external genitalia. Do you have clitoris and labia, or do you have a penis?
Finally, another way of looking at it is gender identity. It's one's own perception of one's sex, and that's how people feel the way they are, either male or female.
And yet, every once in a while, the definitions don't line up. A very small percentage of women have a Y chromosome, and some men don't. Most people with two X chromosomes have female genitalia, but some people with a Y chromosome do too.
We fall over ourselves because of the limitations, the definitions we try to impose. And I think that's precisely the situation that Olympic Committee found itself in.
The Olympic Committee wasn't alone in its quest to define sex. Researchers have been working on this area for many years. For many scientists, it is the area of sex determination that demands the most attention, trying to answer the big question: what makes an embryo develop as male or female?
Prior to 1959, it was actually thought that sex in humans was determined by the number of X chromosomes. And that was simply because that had been shown to be the case in fruit flies, Drosophila melanogaster, back in 1916. And so, since we, like flies, have XY sex chromosomes, it was assumed that we, like flies, determined our sex according to the number of X's.
In 1959, scientists began to test this hypothesis by studying humans with conditions that created chromosomal variations.
One is called Klinefelter syndrome, and these patients have an extra X chromosome. Instead of having 46 chromosomes, they have 47, and they're 47XXY, and they're males.
The Klinefelter study seemed to show that it was not the number of X's that determined sex in humans. Also in 1959, scientists looked at patients with Turner's syndrome, in which one X chromosome is missing. These individuals are known as 45X instead of the usual 46X. And yet, with even just one X chromosome, people with Turner's syndrome are born female.
So what that showed was that no matter how many X's you had, if you had a Y, you developed testes and as a male. But if you didn't have a Y, didn't matter how many X's you had, 1, 2, 3, or 4, you'd develop as a female. So in 1959, it became clear that it's the presence or absence of the Y chromosome that determines testis versus ovary, male or female.
But knowing that the Y makes males wasn't enough. Researchers, including David Page and Eric Verlaine, began a search for what they called the testis determining factor, a single factor sufficient to make the gonads develop as testes.
In fact, what we were looking for is the smallest possible piece of the Y chromosome that was necessary and sufficient to produce a male phenotype in an XX individual. And it took about 30 years to find the smallest ever described portion of the Y chromosome, about 35,000 base pairs. and that was sufficient to give them a male phenotype. Within these 35,000 base pairs, one gene named SRY was discovered in 1990 by the team of Peter Goodfellow in London.
Those 30 years of research from 1959 to 1989 provided the basis for much of our current understanding in the field of sex determination. Once scientists could look beyond the visible chromosomes into the genes and nucleotides that make up the chromosomes, then the underlying mechanism of sex determination became more clear. And when they were able to look more closely, they saw that one chromosome is quite different than the rest. The Y chromosome is unique in a number of ways. While most chromosomes have at least 1,000 genes on them, the Y has fewer than 200. And while most chromosomes have genes involved in a variety of functions and structures, lung and liver and brain and hair, the Y has developed a specialty. Most of the genes are involved in sperm production and sex determination. So where did this unique chromosome come from? For the past several decades, David Page has been trying to answer this question as he has pursued his dream of mapping every gene on the Y.
My whole thinking about the Y chromosome has now been transformed by that understanding that the Y together with the X represent a grand experiment of nature to transform a perfectly matched ordinary pair of chromosomes into this extraordinarily differentiated X and Y chromosomes, something that we understand has unfolded across the last 300 million years of our evolution.
Page has shown that human sex chromosomes evolved from autosomes in our ancient ancestors, back in a time when there were no sex chromosomes.
There are lots of animals that come as male and female that don't have sex chromosomes. In turtles, the sex of an individual embryo is determined by the temperature at which it as an egg incubates. Some species, a little warmer temperature gives you females. Other turtle species, warmer temperature gives you the males.
In bees, it is the number of X's that determines sex. In these haplodiploid organisms, fertilized eggs become female while unfertilized eggs become male. And in some species of fish, like the California sheephead, mature females can change into males. If an alpha male is removed from the group, a female has the ability to turn into a male to replace him.
And we think that what happened about 300 million years ago, a mutation arose on one of a perfectly ordinary pair of chromosomes. That mutation would give rise to what we now know today as SRY.
In 1970, David Page began working on a map of all the genes on the Y. By 1980, he had a rudimentary version, displaying each of the gene families. And with the sequencing of the human genome, Page was finally able to create a complete map of the Y chromosome.
So the mapping of the Y has really gone through several phases, beginning with this rather crude rudimentary map and ending today with a map that is based on a detailed knowledge of the sequence.
The gene on the Y chromosome that gets the most attention is SRY, called the trigger for male sex determination. When the gene was discovered in 1990, many believed the sex determination puzzle had been solved. But now researchers are revealing that puzzle still has many missing pieces.
There were preconceived notions, especially because of the work from Drosophila and from C. elegans, that there would be this nice linear pathway starting out with SRY, and that SRY would then turn on something or turn off something and then turn on the next. There would be this very simple cascade. What both the genetics have told us and the molecular biology and biochemistry have told us that it's complicated. We really need to extend this structure to contain the phosphorylation site. And
Holly Ingram is a professor of endocrinology and a researcher in sex determination at the University of California in San Francisco.
clearly for male sex determination, it's initiated by SRY. But oddly enough, we still don't understand how SRY is talking to the rest of these factors at a real molecular or structural level.
In fact, researchers are now pursuing dozens of genes that may be involved in a complex sex determination pathway, a pathway that seems to carefully balance the genes that push an embryo towards either male or female development.
We enter this new era in molecular biology of sex determination where it's a more subtle dosage of genes, some pro-male, some pro-female, some anti-male, some anti-females, that all interplay with each other rather than a simple linear pathway, which makes it very fascinating but very complicated to study.
In 1952, Alfred Jost conducted a groundbreaking study. Jost removed the ovaries from female rabbit embryos and put the embryos back in the mother's womb. At birth, the rabbits had developed as females, demonstrating that ovaries are not necessary for female development. But when Jost removed the testes from male rabbit embryos, at birth, these rabbits had also developed as females, showing that testes are necessary for male development. From Jost's study, and others, the notion of female by default became the accepted theory in biology, one that has endured for many years. But today, that theory is being challenged.
I think it is ridiculous, actually, to speak of one or the other pathway as default. Both the male pathway and the female pathway are very active and require highly orchestrated, highly integrated sets of events, extremely complicated biochemical cascades that we're only beginning to understand. Truth be known, the prevailing textbook models of how sex differentiation unfolds, those textbook models are extremely biased in favor of the male.
We now know that there must be genes that actively trigger a female pathway. Although we don't know what they are, we know that they must be there. We also know that the female pathway is not necessarily the default pathway because there has been a few cases where you can observe masculinization of a fetus becoming a male newborn without the presence of a Y chromosome.
So our present textbook understanding of sex differentiation in mammals is really a story of how does a male develop. The story of how a female develops has yet to be written. So you got SRY,
Yes.
presumably captured in here.
Ingram, Page, and Verlaine are all pursuing the genes still to be discovered in the sex determination pathway.
Oh, no.
As in the studies that led to the discovery of SRY, studies of intersexuals are central to this work. Intersexuality occurs approximately 1 in every 4,000 births. Newborns are considered intersexual when physicians cannot determine whether they are male or female by examination of their genitals, or when their phenotype does not line up with their chromosomal makeup. In the past, intersexuality was mostly hidden and heavily stigmatized. Today, organizations exist to help end discrimination against intersexuals, and more hospitals are gaining greater sensitivity in handling the issue of intersexuality.
So the baby was born full term, and there was no family history of any kind.
One of my roles at UCLA is to be part of a medical team who is always on call when there is a baby born intersex. Immediately we go to see the baby, talk to the parents, and try to make a precise diagnosis of what is happening.
Yes, they did, so we should go and see the baby.
Okay.
We're able to very quickly give the parents a good idea of what is happening and then help them in the decision-making process.
SRY mutations in intersexuals helped researchers in the 1980s pinpoint this important sex determination gene. But SRY mutations don't explain most cases of intersexuality. In fact, they explain very few.
After the discovery of SRY, we found out that SRY could explain only about 15 to 20% of XY females. In the other cases, SRY had no mutation, which meant that there had to be other genes.
Researchers are now examining a host of promising genes, including Wnt4, SOX9, and DAX1. And though they can't say when, Eric Verlaine, Holly Ingram, and David Page are all confident that in time, we will have a clear map of all the genes involved in both the male and female sex determination pathways. As researchers explore the genetic pathway of sex determination, they're also asking another question. Do genes determine our gender, our own internal sense of being female or male? It's an area that's impossible to study in animals. As far as we know, gender is a uniquely human concept. For many years, it was assumed that gender was mostly a factor of environment. How we are raised determines how we see ourselves. But one classic case seemed to challenge that notion. In 1972, John Money, a sex researcher at Johns Hopkins Medical Center, announced a breakthrough twin study. Janet Reimer had given birth to 2 healthy baby boys, Bruce and Brian. But during circumcision, Bruce's penis was severely damaged, beyond repair. At John Money's suggestion, surgeons performed sex reassignment surgery, constructing female genitalia on the newly named Brenda. Money instructed the parents to raise him as a girl, never revealing his true origins. Brenda was given hormones at the appropriate age to facilitate her development as a female. The study was widely reported in medical literature, detailing how the child was growing up as a well-adjusted girl. Money cited the study repeatedly in his writings. The case seemed to prove that gender could be completely molded by environment. But in fact, Brenda had never felt comfortable as a girl, though she didn't know why. At age 14, Brenda finally learned the truth, and shortly after announced her intention to live as a boy. He told doctors that somehow he had always known he was a boy.
After it was clear that she was very unhappy in his gender reassignment, this hypothesis that nurture could always overcome nature started to be challenged in many circles, and we now know that it's much more complicated. There are so many factors influencing gender that are not only limited to the environment, but also are triggered by hormones, by genetic factors, and maybe by a number of unknown factors that we can't imagine at this point.
I think the case demonstrates that gender identity actually does have biological roots, and that we are not blank slates on which our parents and our environments create us sexually. I'm convinced that gender identity will ultimately be shown to be as biologically based, that is to say, as chemically based, as are the differences in our anatomies.
When the outcome of the case and other similar cases were published, much of John Money's research came under heavy scrutiny. But researchers are still left with the question.
How does this happen? How did she know? I would love to know.
This is one area that we're all extremely fascinated with. Are there genes that contribute to gender identity? And are there genes that will be shared in common that contribute to the plumbing and the gonadal development? Will they also be working in the brain to contribute to gender identity? This was a great case to come to light, and a challenging case, and it challenges us as scientists to try to figure out what is going on with gender identity.
The question of gender identity continues to spark debates in the research world, but some biologists are asking an even more controversial question. Is there a genetic basis for homosexuality?
Homosexuality is an altogether different question. I like to think of it as completely outside of the area of sex determination and gender identity. It's a much more difficult issue to answer. I don't think that we have really good insights into it yet.
My own guess is that there will, in fact, prove ultimately to be genetic roots to sexual preference, as there will prove to be for sexual identity.
If it's true that there is a genetic difference, it will be just different. It's not going to be normal, abnormal. It will be a spectrum of variations at the genetic level.
So these are all pieces of a broad tapestry. Sexual preference, gender identity, sexual anatomy, all of these are pieces of this sexual matrix, which I think will ultimately be understood very much in biological and therefore in chemical terms. But this will require a sort of synthesis and unification that we are far from at present.
These areas of gender identity and sexual orientation can be challenging fields of study as they cross from the world of biology into the social sciences. And even in the more well-understood area of sex determination, this field is still in its infancy. But as we look back, it's clear we've come a long way.
Back in the 50s, we had no idea that even one gene was involved. Now we have a framework of a few genes, so it gives us a springboard to look at other genes and to understand their interactions with the genes that we already know. So we're much further than we were in the 50s. But as usual in science, every new gene unravels more complexity, so the story never seemed to end.
It is most often the first question with any new birth, boy or girl. And while most often the answer is simple, the science underlying the answer can be complex and the social issues surrounding it can be confusing. But as we continue to learn more about the genes involved in sex determination and gain insight into the questions of gender, biology brings us closer to understanding the path that begins with the delicate dance between sperm and egg.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.